For some families, cancer isn't just this random, tragic event. It's a pattern, a shadow that stretches back through generations. And the big question is always why? Well, the answer is often buried deep inside our DNA, a single, tiny typo. And finding it, that is one of the greatest detective stories in modern medicine. I mean, really think about this for a second. Your genome, it's basically a book with over 3 billion letters of code. It's the entire instruction manual for making you. Now, imagine just one single typo in that massive book is causing a devastating disease. How on earth do you even begin to find it? That's the monumental challenge facing scientists who are hunting for the genetic roots of hereditary cancer.
And look, this isn't some abstract thought experiment. This is a real family. This chart, this pedigree, maps out their history with papillary thyroid cancer. For them, finding that one genetic variant could change everything. You see those solid circles on the family tree? Those are the folks we're really focusing on. 2 distantly related family members who both have the disease. The hunt is on.
All right, so to crack a case this huge, you're going to need way more than a magnifying glass. You need a seriously high-tech computational toolkit. So let's meet the hero of our story, a powerful piece of software built to do what seems impossible.
It's called FCVPP-V2. Yeah, it's a mouthful, I know. But think of it as the ultimate genetic sieve. Its whole job is to take this absolute fire hose of genetic data and, through a series of really brilliant steps, whittle it all down to the 1 or 2 clues that actually matter. So how does this digital detective actually work? Well, first it sequences the genomes. Then it catalogs every single genetic variation it finds and checks them against these huge public databases. After that, it tosses out all the common stuff, the background noise. But the real magic, the absolute game changer, is that last step. The pipeline's secret weapon is the family tree itself. And the logic behind it is, well, it's just beautiful in its simplicity. If a variant is really the culprit, it has to be present in the family members with cancer, right? It has to be at the scene of the crime. Any variant that isn't found in all the affected relatives, well, it has a perfect alibi, and it gets thrown out immediately. This one step is the most powerful filter in the entire process.
Okay, so the stage is set. The tools are ready to go. Let's see this thing in action as it takes on a mountain of data to find our one, single, elusive clue. This is where it gets really good.
After the first round of sequencing and some basic filtering, we start with this number, 120,323. Just let that sink in. That is our initial pool of suspects, a genetic haystack of truly epic proportions. So the first command the pipeline executes is the most important one. Check the family tree. The rule is super strict. The suspect variant must be in the 2 distantly related family members with thyroid cancer, and it has to be absent in an unrelated person used as a control. Every single one of those 120,000 variants is about to be interrogated. And boom, just like that, the haystack pretty much vanishes. From over 120,000 suspects, we are suddenly down to just 1,970. In one move, the pipeline got rid of over 98% of the variants. The power of using the family's own genetic story is just astounding. Now the pipeline gets even smarter. It narrows its focus to only the coding parts of the genome, the bits that actually contain instructions for building things. And for every variant it finds there, it calculates something called a CAD score. You can think of it like a threat level. The higher the score, the more likely it is that the variant is a real troublemaker. After applying that filter, only looking at coding variants with a high enough threat level, the list just collapses. From almost 2,000, we are now staring at just 7, the final lineup. Our investigation is getting so close to a single answer.
Okay, we've got our short list, 7 names. Now it's time for the final deep dive forensics. The pipeline uses a whole other set of tools to scrutinize each one, looking for the single variant that stands out from the rest. So here they are, our top suspects in a lineup. The pipeline runs a bunch of other tests to rank them. Think of the deleterious tools column as a panel of different experts voting on whether a variant is harmful. The intolerance tools measure how critical that specific gene is for our health. Now the others have some suspicious traits, but just look at the gene CPXM1. I mean, it's not even close. The CAD score is way higher. It's flagged as dangerous by 9 out of 12 different prediction tools. The data is practically screaming its name. And I really want you to understand, that CAD score of 32, it's not just high, it's insanely high.
It puts this specific genetic variant in the top 0.1% of all possible damaging variants across the entire human genome. This isn't just a suspect anymore. This is our prime suspect. And check this out. Here's another huge piece of evidence. This shows a tiny piece of the CPXM1 protein. See that alignment at the bottom? The code is almost identical in humans, pigs, cats, all the way down to zebrafish. Nature doesn't do that by accident. When a piece of code is kept the same across millions of years of evolution, you better believe it's critically important. Our variant, right there, is a mutation smack in the middle of that preserved region. That is a major, major red flag. But wait, there's more. The background check on this gene turns up some really juicy stuff. CPXM1 is thought to be a tumor suppressor gene. It's also been linked in other studies to breast cancer. And its day job, helping rebuild the physical structure around our cells, is a process that gets hijacked all the time when cancer grows and spreads. The case against CPX1 is looking rock solid.
So it seems like an open and shut case, right? The evidence is overwhelming. We've got our gene. But you know, in science, just like in any great detective story, there's always a twist.
And here it is, the real kicker. Our prime suspect, this exact CPXM1 variant, was found hanging out in a public database of genomes from thousands of perfectly healthy people. So what gives? If this is our killer, why is it walking around free in the general population?
Okay, so this really leaves us with two big possibilities. Is it guilty? It could be what's called a low-penetrance allele. Basically, it loads the genetic gun. It seriously increases the risk, but it might need other things like your environment or other genes to actually pull the trigger. Or is it innocent? It could just be a rare polymorphism, a weird but harmless variation in our code that just happened to show up in this family by chance.
So you might be thinking, well, that's a bust then. But no, not at all. This is actually a huge win. The FCVP-V2 pipeline did its job perfectly. It took an impossibly huge problem and handed scientists one single high-priority target to focus on. An unsolved case file in science isn't a dead end. It's the beginning of a brand new, much more focused investigation.
We started this whole journey looking for a single typo in a 3 billion letter book. And against all odds, we found it. We have the clue. The case might not be officially closed, but the real mystery can finally begin. Doing the lab work, the functional studies, to prove once and for all what role this gene, CPXM1, plays in this family's story. And that is how genetic discovery moves forward.